2019-20 is the golden jubilee year of our college, as in June 2020, college will be completing 50 years to its establishment. As per our vision, our college has played an important role in national development through overall development of Panvel, Uran, Pen and Khalapur tehsils. Our quest for excellence is reflected through some of our outstanding achievements in the recent past. Our college achieved A grade in third cycle of NAC accreditation with CGPA score of 3.18, Best College Award from University of Mumbai, Karmavir Puraskar from our parent institution, successive prizes from Government of Maharashtra in Jagar Janivansa Abhiyan project, ISO 9001-2015 certification from International Accreditation Forum. Friends, college campus blessed with natural beauty, water conservation through college check dam, quality education to 80% backward class students every year. Dear friends, you all know that NAC assessment is based on 5 core values: contribution in national development, fostering global competencies among the students, inculcation of values, use of technology, and quest for excellence. You will agree with me that all academicians are extensively using technology in current situation of lockdown due to COVID-19 pandemic. It reminds me of a famous quotation of Winston Churchill: the pessimist sees difficulty in every opportunity, and the optimist sees opportunity in every difficulty. On the same line, we tried our best, taking into consideration our limitations, to organize webinars for the stakeholders in lockdown period. We successfully organized 6 webinars based on important topics like impact of COVID-19 on Indian and world economy, government measures during COVID-19 pandemic, e-learning as next generation trend in post-COVID-19 era, importance of hydroxychloroquine drug in current scenario of COVID-19, and perspective on health and hygiene amid COVID-19 pandemic. Dr. Pramod Thakur is one of the talented faculty and outstanding researcher of my college, who has 6 published patents to his credit. He has worked as a resource person in many seminars on the topic intellectual property rights. When he conveyed his topic for the webinar, I decided to go one step ahead by organizing a national webinar due to confidence in my technical team, especially my techno-savvy colleagues, Mr. S.L. Gove, Dr. Girish Gund, and Dr. Pramod Thakur. We were surprised to know overwhelming response of the participants for this webinar, which crossed the figure of 700. Our team tackled this issue and upgraded the technology to accommodate all the participants registered for this national webinar. I am confident that all the academicians will contribute to make this webinar a grand success. Before I conclude, once again, I welcome you all for this national webinar. Thank you very much.
Thank you, sir. Thank you so much for motivating us to use this technology and be connected even in this situation. Now, I would like to introduce the speaker of today. It really gives me a great pleasure to introduce the resource person of today's webinar, Dr. Pramod Thakur, a young, enthusiastic scientist in true sense. Being the ex-student of our college, he has brought glories to the college while working in Department of Chemistry as assistant professor. It is a proud feeling to mention here that he has published 5 patents in last 3 years, and recently he has filed 1 more patent. He has completed his PhD in chemistry in 2015 from a renowned university, IICT Hyderabad. He has published 24 research papers in reputed international journals, recognized PhD guide in chemistry. He has received 2 fellowship awards. And not only at college level, he is also working at university level. He is appointed as a coordinator of Raigad district for Avishkar Research Convention. He is also a member of organizing committee of youth festival at university level. So with a very short introduction, I would introduce our today's speaker, Dr. Pramod Thakur sir, and I now request you to begin with the session. Thank you.
Thank you, madam, for warm introduction, and welcome all the dear participants. I am audible to all? Yes. So, in a series of webinars, today I am going to talk on a COVID-19 pandemic from chemistry's perspective. So, last 2 to 3 months, we are stuck in the home only because of the coronavirus, which is known as the SARS-CoV-2. So this SARS-CoV-2 structure we are very much aware nowadays, because every time when we open our phone or when we open the television, we come across this picture. This is a small 150 nanometer molecule which is supposed to be spread from Wuhan, China, who has suffered nearly 5.01 million people in the world and nearly 112K people of India. So we can imagine the harmness caused by this small 150 nanometer molecule. Because of this molecule, since last 3 months, we are stuck in our home due to lockdown to prevent its spread to the community. And this lockdown has created a loss of nearly 9 trillion USD to the economy. So we can imagine the impact of this small 150 nanometer coronavirus on the whole world. So we should look from the chemistry point of view towards this virus, and we will try to cover some aspect of this virus from chemistry's perspective. So if you look closer to this virus, it is named as a coronavirus because it shows a crown like this. So this yellow color part in this figure is known as a crown, which is a spike protein. So there are the 4 major parts of this coronavirus have been identified. Here are the 4 color which we can see in detail like this. So this is the cross-sectional area of this coronavirus. So this yellow color part is known as a spike glycoprotein S. This inside purple color part is a RNA and N protein. This red color part is known as a envelope, and this small blue color part is a hemagglutinin esterase dimer. So if you look at the structure from the cross-sectional point of view, it looks like a crown. That's why it's named as a coronavirus. If you look more closer to this structure, these spike glycoproteins at atomic level have been estimated by the actual image. This is a transition electron microscope image which is taken from the coronavirus from the patient. So this is an actual photograph of a virus which confirms that this is a spherical virus which contains a spike protein, and inside there is a genetic material. This structure of the spike proteins is further confirmed at an atomic level, and it was found that this is the actual protein structure of the spike protein. Okay, this structure was confirmed by using the technology called cryo-electron microscopy. So in this technique, the structure of spike protein was determined, and these results are useful to create a vaccine for this coronavirus disease. When we talk about the coronavirus pandemic from chemistry's point of view, a chemist can see the structure of this coronavirus from chemistry's perspective. So how the chemist will look for this structure? So from the chemist's perspective, this spike protein can be a protein molecule here, which is shown attached to the N-linked protein. So this is a protein molecule which is attached to this sugar molecule. Here we can see the proteins means the polymer of amino acid, where the amino acid are joined together by amide bond. So this spike protein structure from the chemist's point of view can be this type of structure. The second part is a purple color RNA and N protein. So from the chemist's perspective, this RNA is a molecule which is a heterocyclic base attached to the pentose sugar and phosphate molecule. So this is from the chemist's perspective, what is inside the coronavirus RNA, and N protein is a polymer of amino acid. Then the third part is, so from the chemist's point of view, this envelope of the molecule of the coronavirus is made up from a long carbon-carbon chain, which is known as a fatty molecule. And lastly, this small blue color part, which is known as a hemagglutinin esterase dimer. This, from chemistry point of view, it is again a peptide chain, which is known as a proteins, and this is a way by which chemist can look towards this coronavirus structure. So basically, the four part of this coronavirus, a spike glycoproteins is a proteins attached to the glucose. Then inner part, which is known as the genetic material, is a RNA and N protein, which is a N heterocyclic base attached to the pentose sugar and phosphate molecules. Then the envelope red color part is a long saturated hydrocarbon chain, and the small blue color hemagglutinin esterase dimer is a protein. So basically, protein is a polymer of amino acid, and this polymer of amino acid is known as a protein. So from chemistry's perspective, one can see the structure of coronavirus in this way. The structure of coronavirus can be simply understood by this way from the chemistry's perspective. As we know, the coronavirus have a lifetime very short on a different surface. So basically, when we see the coronavirus, the lifetime is different on different surface. Like on steel material, this virus remain for 5 days. On aluminum, 2 to 8 hours. On a glass, 5 days. On a plastic, 2 to 6 days. On PVC, 5 days. Silicone rubber, 5 days. On a surgical hand gloves, 8 hours. Ceramic, 5 days. And Teflon, 5 days. So basically, as we know, the lifetime of this coronavirus is not that much short also. It can live for sufficient longer time. So we must take care of ourselves by cleaning hands and surfaces to stop its spreading, because currently there is no hardcore treatment is available. So because of that, we have to take care that we have to wash the hands and we have to clean the surfaces. So here we are going to see the chemistry behind destroying the coronavirus by cleaning hands and surface. So basically, when we are trying to clean our hand, there is a chemistry is involved in that. So soap and water is used to clean the hand as well as hard surface. So here is the chemical structure of the soap molecule, which is a long hydrocarbon chain at one end, and another end there is a polar group which is dissolved in water. So washing the hand for minimum 20 seconds destroy the coronavirus. So how it destroy the coronavirus? The answer is lies in structure. So just now we detailed structure of coronavirus was seen. What in that we have observed that this red color part, which is known as the envelope of coronavirus, is made up of a hydrocarbon chain. So this is the chemical structure of this red color part, which is known as the envelope. And if you look at the structure of the soap molecule, it is likely looking same. So by the principle of chemistry, like dissolve like, this outer surface of the coronavirus get dissolved in the soap molecule. So by dissolving it, it get destroyed. So once it get destroyed, its harmness is decreased and it is no more active. So this way, washing the hands with soap and water destroy the envelope layer of the coronavirus, and by using the chemical principle we can destroy the coronavirus. So this is the reason why we have to wash the hand with soap and water. This is from chemistry's perspective. Then the second way to destroy the virus is use alcohol hand sanitizer. So in hand sanitizer, basically ethanol and isopropanol are used for both hand and hard surface. So minimum 60% alcohol should be used for hand washing and 75% for the surface cleaning. So how does this hand sanitizer kill the coronavirus or destroy the coronavirus? Again, the reason is same, that in the structure of coronavirus, this envelope layer which is made up from this hydrocarbon chain, this is the organic molecule, and these organic molecules are soluble in the alcohol, as alcohol is a polar solvent. So because of this solubility of this envelope layer of the coronavirus in alcohol, the fatty layers get dissolved and this way protein get damaged. So this way we can look from the chemistry's perspective towards the use of alcohol hand sanitizer in destroying coronavirus. The third way to destroy coronavirus is use of bleach solution. This bleach solution is not for hand, it is for hard surfaces. This bleach is a NaClO, that is a sodium hypochlorite. This percentage of sodium hypochlorite should be minimum 0.1%, so that it will be effective to destroy the coronavirus. So what is the chemistry behind destroying the coronavirus by using a bleach solution? The answer lies in the structure. So as we discussed, this is the structure of coronavirus. Here the yellow color spike protein. This yellow color spike protein contain the proteins and the glucose molecule. So here the protein which is attached to the glucose molecule can contain different oxidizable groups. So this sodium hypochlorite is an oxidant. It undergo the oxidation of the various oxidizable group present in a spike protein and it denature the DNA. So by bleach, which is oxidize and destroy the virus protein and the genetic material, it is used to kill the coronavirus. It is useful for hard surfaces, and the bleach solution should remain at least 10 minutes on a hard surface so that it can be completely oxidize the oxidizable group and deactivate the coronavirus. So this way we can look from the chemistry's perspective when we are talking about the destroying of coronavirus by using bleach solution. The fourth way is use of hydrogen peroxide. Again, the hydrogen peroxide is used for hard surfaces and not recommended for hand cleaning. The hydrogen peroxide concentration should be minimum 0.5% so that it will be effective to destroy the coronavirus. Again, how it works to destroy the coronavirus, the answer lies in this structure. So again, the same reason that the spike protein contain the protein attached to the glucose, which is an oxidizable molecule. There are various groups which are oxidizable groups. So this H2O2 or hydrogen peroxide is an oxidizing reagent. It undergoes the oxidation of these proteins, and it undergoes the oxidation of groups which are available in this spike protein, and because of this it denature the proteins. So basically this hydrogen peroxide destroy the virus proteins and genetic material inside the virus, and it should last for at least 10 minutes on a hard surface so that a virus can be destroyed. So this way there are 4 ways by which we can clean the hand and we can clean the surfaces destroying coronavirus. So next we will see how the chemistry of hand sanitizer is work. So nowadays everyone is aware of hand sanitizer, and everyone is using hand sanitizer, and everyone is now aware about the hygienity. So the production of hand sanitizer is increased tremendously. So what is a hand sanitizer from chemistry's point of view? Hand sanitizer is a mixture of an alcohol. Basically these 3 alcohol, ethanol, propanol and isopropanols are used in a hand sanitizer. Basically the percentage is varies from 60 to 95%. Okay, and the direct relation is that the percentage of alcohol, more will be efficiency, because as we discussed that this alcohol is able to solubilize the fatty layer of coronavirus, which make it destroyed and inactivate. So basically the sanitizer which contain more amount of alcohol, that will be more effective. So basically these are the molecules which are present in a hand sanitizer: benzalkonium chloride and chlorohexidine. Along with this, the glycerol is also used as a moisturizer, because when you apply the hand can get dried, so this glycerol help to moisturize the hand. So this will also help to keep the coronavirus away. Here hydrogen peroxide is also added in the moisturizer because it works as an antibacterial agent, so it can prevent the bacterial contamination in hand sanitizer. So basically how the hand sanitizer work in a similar way, because how the alcohol work in the same way hand sanitizer it work. As we discussed the structure of the coronavirus, the inside is the genetic material and outside is a fatty layer. So the fatty layer structure is a long chain hydrocarbon chain, and this hydrocarbon chain get dissolved with the alcohol. So the alcohol present in a hand sanitizer able to destroy the outside fatty layer, and when the outside fatty layer get destroyed, inside there is a genetic material of coronavirus which get deactivated. So this way this hand sanitizer works. How effective are the hand sanitizer? Because there is a question of hand sanitizer is not proper or they are not useful. So actually they are useful if the percentage of alcohol is proper, means if more than 60% alcohol is present in a hand sanitizer, then only it will be effective. So basically the percentage should be more than 60%, then only it will be able to dissolve the fatty layer of the coronavirus, and it should be 20 seconds. We should have the hand, that time will be sufficient to dissolve the fatty layer by soap molecule. Okay, so we have discussed by this structure, the fatty layer structure contain hydrocarbon. This hydrocarbon can be dissolved in a soap molecule or in a alcohol present in a hand wash molecule. So basically this what we have discussed about the molecules and hand sanitizer, how the coronavirus can be destroyed by using these chemical products. Apart from this, if somebody is get positively tested for coronavirus, or if somebody has the symptom of the coronavirus disease, then he should go for the test center and look for the test. So basically the test center will be look like this, where the sample collection takes place, and this is a glove box. This is a healthcare person who is collecting the sample without coming in contact with the suspect or the patients. So this type of test centers are available in our country to collect the sample, and the sample collection is done from these two ways. It can be collected from the nose or it can be collected from the throat, and then it can be treated in a PCR machine to get the data. Currently, the test which is used for the confirmation of presence of coronavirus is a RT-PCR test. So we will see the chemistry behind this corona test, how the chemistry is working in this test. So this RT-PCR test, which is known as a reverse transcript polymerase chain reaction. So this RT-PCR test is a technique currently used to detect the presence of coronavirus. So what is this RT-PCR test? It is a reverse transcription polymerase chain reaction. Is a technique that first uses reverse transcription to convert the extracted RNA from coronavirus into DNA. We have thus discussed the structure of coronavirus. Inside the fatty layer, there is a genetic material of coronavirus, which is known as a RNA. So this RNA is extracted from this coronavirus, and then it is transformed into DNA, and then uses this PCR, means polymerase chain reaction technique, to amplify the piece of resulting DNA to create number of more copies of this DNA, which will be sufficient to detect the presence of coronavirus. So this is the test behind the detection of coronavirus. And if you look the test in detail, it is shown here. When this cell from the coronavirus is extracted, it will give the RNA. This RNA from this coronavirus will be purified, and this purified RNA will undergo the first strand synthesis to synthesize the DNA, and this DNA then undergo the PCR technique to multiply the DNA or make its copies, and it will amplify the PCR, and it will create large number of copies of the DNA, and this large number of copies of the DNA can be detected by using the particular technique. Okay, so we'll see much detail about this technique. So how this current test work, and what is the chemistry behind this current test of coronavirus. So first step, a swab is taken of the inside of patient nose or back of their throat. This sample is then sent to the lab. So this is the way by which sample is collected, and this is the coronavirus which is collected from the nose or back of the throat. Here inside there is a genetic material, this central part. Okay, in this central part, this genetic material is a virus RNA. Okay, so this virus RNA is extracted in the first step. In the second step, the RNA of the virus is extracted and purified, and enzyme reverse transcriptase convert the RNA to DNA. So this stage is very important. In the presence of virus genetic material RNA, the reverse transcriptase convert it into DNA. That's why this test is known as a RT-PCR. So this RT stands for reverse transcriptase. This is a key step in this test. So in this step, this RNA of the virus get converted into DNA molecule. Then in the third step, this DNA, which is prepared by RT-PCR method, is mixed with the primer, and then it is repeatedly heated and cooling. DNA with this primer and DNA building enzyme make millions of copies of this DNA. So what happened here, when the DNA is heated with the primer and it creates number of copies, and when it is treated with the dye, now here the dye which is used is a cyber green dye. This is the structure of the dye which is used in this RT-PCR test. When this dye is bind to this copied DNA, as the number of DNA copies will be more, there will be more fluorescent. So this fluorescent dye makes bind to the virus DNA. Here red color is a dye which is bind to the virus DNA as it copied. So binding make them give off a more light, which used to confirm the presence of virus in the sample. So basically, when this dye get attached to the DNA, the resulting DNA dye complex blue light 497 nanometer and it emits the green light at 520 nanometer. So as the number of DNA copies increases, there will be the more fluorescence and this will detect the presence of this coronavirus. So basically how the positive and negative tests are confirmed. So increases as more copies of the DNA are produced. So we are going to get this type of graph if the coronavirus is present, because the fluorescence intensity will increase with increasing number of cycles. So if there is no coronavirus, there will be no increase in the fluorescence intensity as number of cycle is increases. So this type of graph shows the presence of the coronavirus, because this type of graph is only present when there is a presence of coronavirus. So this is what the chemistry behind the coronavirus test, but this test have some limitations, or it test have some drawbacks that we'll see. That what are the issues with this testing. First thing is the reagent use. There is a high demand and issue with the reagent have delayed a testing in some countries. At currently, this only the tests which are used for detection of coronavirus throughout the world. So there is a shortage of these chemicals used in this test. That's why there is a delay in some countries. So there should be the alternative test. Second is that it is a time consuming. So it takes few hours to get a result from the test, limiting how many tests can be done. So because of that, there are very limited tests can be done in a day. So because it is a time consuming, sometimes we are also hear in the news that some person first get positive, then it tailed as a negative report. Sometime a negative person again tested come a positive. The reason is that in some cases sample degradation or contamination can affect the result. So the key step in this test is the isolation of RNA material from the coronavirus. So in that stage, if the degradation of RNA takes place, then the test can give a different result. So these are the some issues with the current RT-PCR test. So there must be a different test which is known as a future test. So what is the future test for detection of the coronavirus? The future test should be based on a measurement of antibodies. Okay, so basically when infection takes place in a person of coronavirus, the body creates antibodies to neutralize the effect of this coronavirus, and these antibodies remain in some time in our body. So the test should be formulated which will able to determine these antibodies, so that we can able to determine the test more precisely. Again, the drawback of this current RT-PCR test is that it is only diagnosing an infection, but they can't tell us if someone has had it and then it recovered. So basically this test based on these antibodies will be the better test for the detection of this detection of this coronavirus disease. Okay. So what if somebody get tested positive of coronavirus, then the next step will be look for the treatment for this coronavirus to get cure. So basically when we look for the treatment of this coronavirus, in media we are completely known about the word hydrochloroquine. Here we can see how the importance of this hydrochloroquine. The two big powers have come in the same line due to this hydroquinone. So this hydroquinone, then this Trump and this PM Modi. So two big powers are connected with this hydrochloroquine drug. So this is what we can say, this importance of chemist in the current COVID-19 pandemic. This person we have always seen currently in media talking about the hydrochloroquine and India. There are a lot of discussion on the news in the last few months about this coronavirus, political debates, many discussion about this hydroxychloroquine drug. lot of discussion on a Twitter handle about this hydroxychloroquine drug. So actually, what is this hydroxychloroquine? This is a small tablets which is used in a 20 milligram form, basically prepared by the Cipla and in marketed by the name of Plaquenil. Okay, so basically this hydrochloroquine tablet, this is the structure of this hydrochloroquine. So this is the chemical structure of the hydrochloroquine, and if the IUPAC name of this structure is RS247 chloroquinoline 4-il aminopentyl ethyl aminoethanol. So if you look closely, it is a quinoline ring, and this is a diamine substituent. So a simple structure of this hydrochloroquine. So basically this hydrochloroquine is a drug that has been used in decades for treatment of hydroxychloroquine is a drug that has been used for decades in treatment of malaria. The brand name of hydroxychloroquine is a Plaquenil, and it is also known as a hydroxychloroquine sulfate. It is listed by World Health Organization as one of the most essential drugs, safest and most effective medicine needed in a healthcare system. So if you look carefully, actually this hydroxychloroquine is a drug which is used for the treatment of malaria. Then what is its consequence in this COVID-19? Why it is become so much famous during this COVID-19 pandemic? Because during this COVID-19 pandemic, the hydroxychloroquine become very important. The reason is that this has emerged as a most promising drug to treat COVID-19 patients. US FDA has identified hydroxychloroquine, which is abbreviated as a HCQ, as a possible treatment for COVID-19. And because of the significance of this hydroxychloroquine, this drug become very important in this pandemic, and it emerged as a most promising drug. That's why the US president is talking about this hydroxychloroquine every time. So because of this important drug, the clinical trials have already started in the US with 2000 patients, and it has shown the promising results. So there is definitely hope with this hydroxychloroquine drug. This current demand of this hydroxychloroquine now the increased tremendously, because since US FDA has identified hydrochloroquine as a possible treatment for this COVID-19, the demand for this hydroxychloroquine is increased drastically. Before the outbreak of COVID-19, the domestic requirement from this hydrochloroquine was around 20 lakh tablets per month. But see, now India producing 20 crore tablets of 200 milligram HCQ every month, and which can be scaled up to 30 to 35 crore tablet every month. So you can see how there is a tremendous fold increase in the demand of hydroxychloroquine. It clearly indicates that how this hydroxychloroquine drug is important in the current scenario of the COVID-19. So basically, India have enough stock of this hydroxychloroquine, and in the last 2 months, India has supplied nearly 120 countries this hydroxychloroquine drug. And because of this hydroxychloroquine drug, India become important country to the whole world in this COVID-19 pandemic, and India has taken the advantage of this, and India got chance to play the medical diplomacy with the developed countries because of this hydrochloroquine drug. So the chemists have prepared this hydroxychloroquine drug, and this drug become important in this current scenario. So we must proud ourselves being a chemist, because chemists come with the solution for the coronavirus patients. So this hydroxychloroquine drug, the look very simple, and if you look the synthetic part, it is only the 3-step synthesis of this hydroxychloroquine. The first step is the formation of carbon-nitrogen bond by simply nucleophilic displacement to form this molecule. Then next step is a reductive amination. So reductive amination takes place in the presence of hydrogen, ammonia and radionickel to form this molecule, and then it gets coupled with this quinoline ring to form this hydroxychloroquine. So this three-step molecule is become very important or magic drug in this current scenario of the COVID-19. So basically, when we treat this drug or when we give this drug to the patient, the results shown that a significant improvement in a patient health. So basically, when we look for this hydroxychloroquine drug, it should be given under observation and it should be given only in a prescribed manner. So this is what about if you get the positive tested or if you get infected with the coronavirus. But if you try to prevent yourself for getting infected with the coronavirus, then in the current scenario, there is a weapon which can save us is the face mask. So basically, what we are using today for taking away from this coronavirus is a face mask. So we can treat this as a today's weapon against COVID-19. Okay. So in the market, there are many types of face masks are available. I come across a very interesting article which is published in ACS Nano regarding this efficiency of the face mask. So in the chemistry of face mask, the recent article was published in ACS Nano in April 21, 2020, which have tested the aerosol filtration efficiency of common fabric used in respiratory cloth mask. So in this excellent paper, they have studied the various parameters of the face mask. They have studied the multifunctional face mask, various conventional face mask, multi-layer face mask, and they come with very interesting conclusion about the face mask. So they have actually, this is the graphical abstract of this paper, that first layer filter the aerosol particle and then mechanical filtration give the pure air. So this way the face mask, which is a multi-layer, is purify the air and avoid the particulates. So what their conclusion was in that paper, they have carried out these studies for several common fabrics, including cotton, silk, chiffon, flannel, various synthetic and their combination. What they have observed, although the filtration efficiency for various fabric when a single layer was used range from 5 to 80% and 5 to 95% for a particle less than 300 nanometer and particle more than 300 nanometer. So efficiency was more for this particle which is having size more than 300 nanometer. Further, what they have observed, the efficiency improved when multiple layers were used and when using the specific combination of different fabrics. So this is what important, that you should use the multi-layer mask. So this research have confirmed the efficiency is more with the multi-layer mask. Further, what they have observed, the filtration efficiencies of the hybrid means multi-layer such as cotton silk, cotton chiffon and cotton flannel was more than 80% for particle less than 300 nanometer and more than 90% for particle greater than 300 nanometer. They speculate that the enhanced performance of the hybrids is likely due to the combined effect of mechanical and electrostatical based filtration. So this is also important observation, that multi-layer mask why should be used, because it gives the combined effect of this mechanical and electrostatic based filtration. The cotton, the most widely used material for cloth mask, perform better. This is very important thing to note, that any cotton material in our home that can be used to mask our face and can make significant difference in the filtration efficiencies. Further, their observation was, the studies also implies that the gap, this is actually very important observation, as caused by the improper fit of the mask. So if mask is not properly fitted, then what can be caused? It can result in over 60% decrease in a filtration efficiency. So this is very important point which they have noted, that the mask should be properly fit. So this type of observation implying the need for future cloth mask design studies to make into account the issues of fit and leakage while allowing the exhaled air vent efficiently. So basically what they have highlighted here, that mask should be properly fit, and it should be designed in such a way that it should be fit properly, because there can be a 60% decrease in a filtration efficiency, and that cannot give the protection from this particulates as well as this coronavirus. Overall, what was their observation? That overall they find that combination of various commonly available fabrics used in cloth masks can potentially provide significant protection against the transmission of aerosol particles. So this was very interesting research which was published recently in this COVID-19 pandemic related to this efficiency of face mask. And I found that it should be good to share this information with this participant. This paper is available in ACS Nano Journal. One can read the paper in detail if they are more interested in this type of work. So what it suggested, that when you go outside the home, you should always wear the mask, and you should always maintain the social distancing, because when you are wearing the mask and when you are maintaining the social distancing, there is a maximum possibility that you will get less infected or not infected with this coronavirus disease. So what is the science behind the social distancing that we are going to see here? The science behind social distancing, which we are nowadays following in many parts of the country, people are following this social distancing as a restriction as provided by the government, that everyone should follow the social distancing. So from the chemistry's point of view, or from the science point of view, what is the science behind this social distancing? Basically, a coronavirus travel in droplets expelled from the original host via sneezing, coughing, or just speaking. So how the coronavirus come outside the host? By these three processes: sneezing, coughing, and speaking. These droplets will either evaporate quickly in air or be dragged by gravity down the earth. Now see what happens. If the size of droplet is small, they can evaporate in the atmosphere once they come out. If the size of droplet is more, they can be dragged by gravity down to the ground. So this is very important observation. The majority of those droplets will land within 1 to 2 meter, because size of virus is 150 nanometer, which form a droplets of nearly size of 1 micrometer. So this is very important, that the droplets which is coming out from the host will land within 1 to 2 meter, because it form a droplets of a size 1 micrometer. The virus can only survive for limited time outside of the host and need to get next host quickly. So this is what important thing, that it cannot remain for longer time on outside the host, and it required immediately the new host. If a host is standing within a range of a travel by the virus containing droplets, then they have a high probability of catching the droplets and becoming infected. So that is the reason that you should maintain the gap of 1 or 2 meter in social distancing. Here we can see the projection, the infected person, and here we'll see the coronavirus try to come outside by the process of sneezing, coughing, or just speaking. So the size of virus is nearly 150 nanometer, and the droplet size is 1 micrometer. So basically, if larger the size, they will remain on a ground at a distance of 1 meter due to the gravity effect. So larger the size, it will fall close. Particle size or droplet size decreases, the distance will be increased. So basically the maximum danger zone is a 1 meter. That's why it is suggested you should maintain the social distancing of at least 1 meter. Okay, so because maximum threat is in 1 meter, there is a further threat up to 1.5 meter or 2 meter. Okay, but the threat will be very less, because the small size droplets can evaporate in the air. Okay, but this large size droplet, which is formed because of the coronavirus inside, which is having 150 nanometer, it become large size of 1 micrometer, and it fall in a range of 1 meter. So this is the reason why one should follow the social distancing, and you should keep away from this coronavirus disease. So this is what important about the social distancing. And when we talk about the social distancing, we should always wear a face mask when we go outside the home, or when we are not in the home, we should always wear a face mask. So there is interesting research which is going on in a various aspect in this COVID-19 pandemic, and we must proud as a chemist that recently renowned academician from the University of Mumbai has come forward to do some research to combat this COVID-19 pandemic and help the or save the lives. Okay. So I am going to highlight the recent achievement of Department of Chemistry, University of Mumbai, who have come forward to do and very useful research to combat this COVID-19 pandemic. So this is the person who is working for combat COVID-19, Dr. Vishwanath Patil, Associate Professor, Department of Chemistry, University of Mumbai. During this COVID-19 pandemic, we are sitting in the home to stay away from this COVID-19 virus and keep us safe. This faculty member of Department of Chemistry is working hard to come up with the solution to combat the COVID-19 pandemic, and he has done very important research in this COVID-19 pandemic, and he has come up with the useful solution. So this research include, he has developed a nanotech based antiviral coatings for application on PPE and N95 face mask to kill the virus. So this is very, very important research which is done in the current scenario by chemistry faculty member. That's why I purposely mention this contribution in this presentation, because when we look for the COVID-19 from chemistry's perspective, we must highlight the hard work of the chemist during this COVID-19 pandemic. The coatings which is prepared are based on the specially designed medical grade polymer and nanoparticles of certain metal. So sir has developed an antiviral coatings which is prepared from medical grade polymer and nanoparticle of certain metals. The coatings can be synthesized within 4 hours and dried within 15 minutes after the application. It will prevent any kind of infection and help save lives of medical staff and frontier workers in the current COVID-19 pandemic. So this, I can say, very important contribution from the chemistry faculty of the University of Mumbai in this current COVID-19 pandemic. So we must proud as a chemist that chemistry peoples are working hard to find us on this COVID-19 pandemic. These are these work which is going on in his lab and which are already tested and submitted to NIH, that is National Institute of Virology, Pune, for further process. This is the person who is working in his lab hard during this COVID-19 pandemic. This is the coating which they have made to apply on the PPE and face mask. So this is very important work done by chemists during this recent COVID-19 pandemic. These are the various newspaper highlighting his research and giving the appreciation. Dogs may soon be dressed to kill virus thanks to nanotech research at varsity. Mumbai University develops nanotech based coating to kill virus on PPE. Then some newspaper in Marathi, PPE kit N95 mask वरील कोरोना विषाणू नष्ट होणार, डॉक्टर होणार अधिक सुरक्षित, अँटीव्हायरल नॅनो कोटिंग्स कोरोना विषाणू नष्ट होणार, मुंबई विद्यापीठात अँटीव्हायरल नॅनो कोटिंग्स तयार, मुंबई विद्यापीठ व स्वच्छ अलायन्स एकत्रित संशोधन, मास्क वरील विषाणू नष्ट करणे शक्य, अशा वेगवेगळ्या न्यूजपेपर मध्ये हे रिसर्च वर्क हायलाइट झालेलं आहे. These are the links where one can read detail about this research and other more references of this presentation. In this COVID-19 pandemic, when we are sitting in the home, there are some positive things which are happening. Roads are empty, because of that there is very less pollution. There is data showing that there is a less than there is a decrease in nearly 40% pollution during this COVID-19 pandemic or during this lockdown period. If people are not on a road, the place is taken by animals. So we look it very fantastic that animals on road, but this is our, we soon want to go on the road, we soon want to go to our workplace, and we soon want to get out of this COVID-19 pandemic. I take this opportunity to thanks Honorable Principal Dr. Ganesh Thakur Sahib, ex-secretary Ray Shikshan Sanstha Satara, and Principal Mahatma Phule Arts Science Commerce College Panvel for giving this opportunity to give the webinar on this current topic. I also thankful to my head of the department, Mr. J.J. Korane, for giving me opportunity and encouraging me. I am thankful to my NAC coordinator, Mr. S.L. Gove, for arranging the technical facilities for this webinar, and I am thankful to Dr. Vishwanath Patil, Associate Professor, Department of Chemistry, for guiding me for and giving information about his current research. So till then, I wish you all stay home, stay safe, and keep e-learning. Thank you for attending this webinar.
Thank you, sir. So with this, we have come to a the end of this very informative and very knowledgeable session. So thank you so much for telling us about the different products which can be used for sanitization and cleaning our hands, and what precautions we should take while using these chemicals or hand sanitizers. Thank you so much.